Hello everyone, in this video we are going to discuss about Corynebacterium diphtheria. So Corynebacterium diphtheria, it is a gram-positive organism which is a club-shaped rod. So it is arranged in a specific pattern called as Chinese letter pattern or palisades. Next is it contains metachromatic granules which can be seen on Albert stain. So we can see in this picture we can see bipolar granules which can be seen on Albert stain. And this picture is showing gram-positive rod-shaped organism. So coming to the virulence factor, how this organism will cause the disease is these Corynebacterium diphtheria will have a toxin-producing organisms. Only the toxin-producing organisms will cause the disease. These have specific tox gene. So the toxin of the Corynebacterium diphtheria will have two parts, that is part A and part B. A stands for active part and B stands for binding part. So what happens is, first this B part will bind to the HB-EGF. So this HB-EGF is nothing but heparin binding epidermal growth factor. So next, after binding to this, the B part will bind to this HB-EGF, and this is taken inside the cell through clathrin mediated endocytosis. So inside the cell it is being taken, so that's why it is endocytosis. So in high pH levels, that is alkaline pH, what happens is the protease will partially cleave this diphtheria toxin subunit bond, that is this B and A bonds will be broken, and in low pH, that is acidic pH, will trigger the translocation of this A subunit. So this A subunit, which is the active part, will actually cause the disease. So what happens is this A domain, the active part, will catalyze the ribosylation of the EF-2, and this causes the modification in the EF-2, causing inhibition of the protein synthesis. So the first what happens is the DT bind to the HB-EGF, and there will be clathrin mediated endocytosis, and protease partially cleave the DT subunit bond, and acidic nature will trigger the translocation of the A subunit, and A domain catalyzes the ribosylation of the EF-2, and this causes ribose modification and inhibition of the protein synthesis, which finally leads to cell death. So in gist, the toxin will contain two parts, A part and B part. A is active, B is binding. This inhibits, the A part will inhibit the protein synthesis, causing cell death, and this causes various clinical features which we will see in the disease, that is pseudomembrane formation. Pseudomembrane is nothing but fibrin with dead cells, and heart damage which causes myocarditis, and nerve damage causing neuropathies. So these, coming to the clinical features, as we already know, these causes pseudomembrane formation in the throat, which is grey and necrotic, which is nothing but fibrin with dead cells, and there will be cervical lymphadenopathy which causes appearance of bull neck appearance, and the complications include myocarditis, arrhythmias and neuropathy. So coming to the diagnosis, how we will diagnose this Corynebacterium diphtheria. First is gram staining, which we will see, where we will see the gram positive rods, and we can see the metachromatic granules on Albert stain. And coming to the agar, that is there is a black colony formation on tellurite agar, and we can also see the growth in the Loeffler's medium, and the main two tests are the Elek test and the Schick test. So coming to the Elek test, it is used to check if there is a diphtheria toxin production. So this works in the principle of immunodiffusion. So on an agar plate, what we will do is we will place an antitoxin soaked filter paper. To right angle to this, we will place 3 strips. First strip is toxigenic Corynebacterium diphtheria, which is a positive control, and the last strip is the non-toxigenic Corynebacterium diphtheria, which is a negative control, and the middle strip is the test which we are doing for the person. So it is unknown. So if there is a precipitation line, which we will see because of the toxin-antitoxin interaction, there will be precipitation line. So with the positive control, there will be precipitation line. If also with the unknown control, we are seeing the unknown strip, we are also seeing the precipitation line, it indicates that there is a toxin production of the strip. So this is indicative of there is a toxigenic production in the species which is actually being tested. So next test is the Schick test. So it tests for immunity and susceptibility. So what we will do is 0.1 ml of the toxin, which is both activated and inactivated. Activated is directly and inactivated is which is heated. So this one activated is injected intradermally on the right forearm, for suppose, and on the left forearm we will inject intradermally the inactivated one. So we will take time of 24 to 48 hours and read the readings after this time. So if there is no redness in any of the arm, it is negative. If there is redness in the right arm, that is a activated thing, it is positive. And if it is redness is seen in both the arms, both sides, it is hypersensitivity reaction of the error. So so if the person is having redness on the activated side, it indicates that the person is susceptible, and if there is redness on both sides, there is hypersensitivity. So coming to the treatment, first we have to immediately give antitoxin, which is immediately we give antitoxin, and penicillin or erythromycin can be given, and DPT vaccine should be given. So these are the important points in Corynebacterium diphtheriae.